Good afternoon.
Yeah, good afternoon, Dr. Owosho.
Good afternoon, Dr. Amshiti. Good afternoon, everyone. My name is Adepitan Owosho, and today I'll be giving a talk on a very minor research work that we've been able to do here at AT Still University with the Missouri School of Dentistry and also with my external collaborators from the University of Pittsburgh and also the Air Force Base in San Antonio. So today my presentation would be on the molecular characterization and clinicopathologic features of secretory carcinoma of minor salivary glands. So just before I get into that particular topic, I would like to introduce you into the world of salivary gland tumors in a very brief introduction. So this is the paper that we were able to work on and we were able to publish. And this is currently in press. This was published in the Journal of Oral Surgery, Oral Medicine, Oral Pathology, and Oral Radiology. This article was accepted for publication on the 1st of April. And this is what I'll be talking to you about. So talking about saliva as an introduction, generally, approximately 1.5 liters of saliva is produced every day. Even when you don't notice that you're producing saliva, every single day we tend to produce about 1.5 liters of saliva. And on the resting rate of saliva production, when you're just resting, you're not doing anything, you're not eating, like right now, you're sitting down listening to this presentation, the rate of saliva production is about 0.5 mils per minute. Now, when this saliva production is stimulated, a very good example would be when you're eating or when you smell a very nice aroma of food and you tend to start to salivate, that's when your saliva is being stimulated. The rate at which this saliva is being produced is 10 times that of the resting rate. So that would then make it 5 mils every minute, the rate at which the saliva will be produced. And as we all know, saliva plays many vital roles. It helps us with tasting, with speech, with swallowing, most importantly with irrigation and cleansing, also helps as a lubricant. It also tends to have its own antibacterial mechanism in saliva, such as the immunoglobulin A and other antibacterial factor that has been seen in saliva. And as a dentist, one of the most important vital role of saliva to a dentist is the buffering capacity of saliva. Saliva tends to contain a lot of buffers, most commonly the bicarbonates. The phosphate saliva is also one of the buffering capacities of buffering factors that we tend to see within the saliva. However, the most common one is the presence of the bicarbonates in saliva. So what are the sources of saliva? Basically, the sources of saliva can be broken down into two major categories. We have the major salivary glands and the minor salivary glands. The major salivary glands are the submandibular, parotid, and sublingual salivary glands. And the minor salivary glands, they're just about 600 to 1,000 minor salivary glands in the oral cavity. They are located on the buccal mucosa, which is the lining of the cheek, of the cheek in the mouth, the labial mucosa, which is the mucosa around the lips, both the upper and lower lips. The palatal mucosa is the mucosa that you feel on the roof of your mouth. If you put your tongue over the roof of your mouth, the mucosa you're touching right there with your tongue is the palatal mucosa. The lingual mucosa, this is the mucosa that lines the tongue, the ventral surface of the tongue, and also minor salivary gland can also be seen at the base of the tongue. The majority of the salivary glands that are seen in minor salivary glands are considered to be of the mucinous acinine tissue, so therefore they tend to produce a lot of mucinous saliva. Looking at the microanatomy of the salivary glands, you can see here's a tissue section, a photomicrograph of a parotid gland, and you can see mainly you see more of the serous acini of the salivary gland, and more importantly is the ductal structure within the salivary gland. So all the way from the area of the acini, you have the the caniculus to the intercalated duct, to the striated duct, from the striated duct communicating to the excretory duct, and from the excretory ducts coming together to the main excretory duct. For the parotid gland, that main excretory duct is known as the Stenson's duct. For the submandibular, is known as the Wharton's duct. So you have all this collection of ducts coming together into the main excretory ducts, and thereby saliva is then being secreted out. It's very important for us to know that saliva production or saliva secretion, it's mainly of a neural control. It's not under any form of hormonal influence. So the fact that you're able to produce saliva or feel saliva in your mouth is because there's a neural control that helps in that secretion of saliva. And at the very end of the acini of that saliva, you have the myoepithelial cells around the acinus. And what the myoepithelial cells, what they do is that basically they help to squeeze this acini area, and by squeezing it, the saliva contents within those areas are able to be expressed into the duct, and thereby being expressed out into the oral cavity. So this is a basic microanatomy of the salivary glands. So talking about salivary gland neoplasms, salivary gland neoplasms in general are not common. They are not common compared to other forms of cancers in the body. However, they make up about 5 to 7% of all head and neck tumors in general. Salivary gland tumors can be broadly categorized according to their presumed origin of differentiation as follows. For example, you can categorize them based on their origin. So we could have salivary gland tumors that will fall under the category of the ductal acini. That is, these are tumors that arise from the ducts. The ducts I just mentioned, intercalated duct, striated duct, excretory duct. So if you have a tumor arising from the duct and the acini portion of the salivary gland, those tumors will fall under the category of the ductal acini. Then we have the biphasic tumors that are considered to be biphasic. And they are said to be biphasic because they are made or they are produced from a combination of both ductal and myoepithelial components. So remember the ducts from the microanatomy. I remember the myoepithelial components, the myoepithelial cells that kind of wraps around the acini, that basically squeezes the acini for saliva to be expressed out into the duct. So if you have a tumor that is arising both from the ductal system and the myoepithelial cells, they are considered to be biphasic, and there are categories of tumors that falls into that category. We have tumors that are purely myoepithelial. These are tumors that purely arise from myoepithelial cells. They are purely myoepithelial. We also have tumors that are considered to be excretory duct phenotype. Remember, in the ductal system, from the caniculus to the intercalated duct, to the striated duct, to the excretory duct, if you have tumors arising from the excretory duct specifically, then those will be put under the category of the excretory duct phenotype. And the last category will be miscellaneous or indeterminate origin. So let's look at a couple of examples under each of these categories. So looking at this again, just to show you the microanatomy. So talking about the various areas, we talked about the ductal acini, tumors arise from the duct and from the acini. We talked about the purely myoepithelial cells. We talked about the biphasic, those that are a mixture of ductal and myoepithelial components. Then we talked about the excretory duct phenotype, those that are definitely only from the excretory duct. So again, this kind of shows you where the different tumors could arise from the microanatomy of a salivary gland. So here's an example. Here are examples to see. In your ductal acini, the following are the examples. If you want to, basically, we can categorize tumors into benign and malignant based on their clinical features. So looking at the benign category of tumors that arise from the ductal acini, very two common examples are the canonicular adenoma, oncocytoma. Also, you have under the malignant category, we have the salivary duct carcinoma, secretory carcinoma, which is what we'll be talking about today. Our talk is mainly on secretory carcinoma. Other examples would be your polymorphous adenocarcinoma, arsenic cell carcinoma, intraductal carcinoma, and adenocarcinoma NOS. So all these tumors are examples of salivary gland tumors that are considered to be ductal acini. Under the biphasic categories, We can also categorize them into benign and malignant. For the benign example, the most common tumor, which, if you're familiar with salivary gland tumors, pleomorphic adenoma, is considered to be a biphasic tumor. Basal cell adenoma, Wharton tumor. The malignant components of this are adenocystic carcinoma, epithelial-myepithelial carcinoma, basal cell adenocarcinoma, and carcinoma X pleomorphic adenoma. Very briefly, I would like to talk about these very common entities, the ones I have in bold, so as to give you a refresher and to give you a broad understanding of salivary gland tumors. So we'll talk about pleomorphic adenoma. Pleomorphic adenoma is the most common salivary gland tumor. It's also the most common benign salivary gland tumor. So it's the most common salivary gland tumor, either benign or malignant. And when you consider benign tumors, it's also the most common benign salivary gland tumor. Now, when we talk about salivary gland tumors within the oral cavity, pleomorphic adenoma is also the most common. Typically, the most common location for pleomorphic adenoma is the parotid glands. That's the gland that you tend to feel around your cheek area, the parotid gland. That's the most common location for pleomorphic adenoma. However, when you consider intraoral salivary gland tumors, the most common location for pleomorphic adenoma would be the palate. Again, as I said earlier on, pleomorphic adenoma is a biphasic tumor. That is, it has both the ductal epithelial component and also the myepithelial component. And we have this epithelial-myepithelial component. We tend to see it, both of them are in a chondroid myxoid stroma. The epithelial cells typically would form ducts, while the myepithelial cell types, there are four different types. As pathologists, we tend to call them either plasmacytoid, epithelioid, clear, and spindle. And I will show you examples of why we do call them that. And the known genetic alteration that has been seen in pleomorphic adenoma is the PLAG1. PLAG1 is the most common genetic alteration that is known to be present in pleomorphic adenoma. So when we talk about the myepithelial cells, we said there are four different types. The first type is this plasmacytoid. It's called plasmacytoid because those cells individually resembles plasma cells. That's why they're called plasmacytoid. The other category is the epithelioid. They look squamoid, so they are said to be epithelioid. Next category will be your clear cells. And as you can see here, you can see that the individual cytoplasm around the nucleus of these cells are clear. So they are called clear cells. And the last category is what we call the spindle. So these are all the various types of myepithelial cell types that we could see in pleomorphic adenoma. Talking about the stroma is usually of a myxoid stroma, or it could actually look like a cartilage. That is why it's called a chondroid stroma. So sometimes you could see a pleomorphic adenoma that will mainly contain a myxoid stroma, or it could look like a cartilage, and it's called a chondroid stroma. In some situations, you could actually have a mixture of both myxoid and chondroid stroma. So that will be a myxochondroid stroma or a chondromyxoid stroma in your pleomorphic adenoma. The stains I'm showing you here are basically showing you, they are basically highlighting the myepithelial cells. And this is kind of very important because if you take a very close look at this, you can actually see that the ductal epithelial cells are not staining, are not positive for this stain. However, the myepithelial cells are positive. And that kind of shows you the biphasic tumor we're talking about, whereby you have both ductal cells and myepithelial cells. And the ductal cells, as you can see here, they are negative. They're not staining for this stain, while this P63 stain is basically highlighting the myepithelial cells. The same also applies with the use of S100 stain also. Another common tumor we'll briefly talk about is adenocystic carcinoma. It's also a fairly common tumor in salivary gland tumor. In this situation, this is a malignant salivary gland tumor. It can be seen in the parotid gland, submandibular gland, and also in the palate. It's slow-growing, indolent tumor. However, it's known to be associated with perineural invasion. What does this mean? It means that this is a kind of tumor that tends to track along the nerves. So the mode in which this tumor tends to spread most of the time is through a local spread, and it tends to wrap around nerves. And you can imagine the amount of nerves in the head and neck region. And because of this particular preference for perineural invasion, this tumor can spread all the way to the nervous system. It can spread all the way to the brain. It can spread all the way to the base of the skull by just tracking along the nerves. So it's very, very distinctive that adenocystic carcinoma has a great preference for perineural invasion. It's also a biphasic tumor. You can see the epithelial ductal cells and myoepithelial cells. There are 3 morphologic subtypes. We have the solid, cribriform, and tubular. And also the genetic alteration seen in adenocystic carcinoma is the MIB-NIF gene translocation. Here's a solid pattern of adenocystic carcinoma. Here's the cribriform pattern. The cribriform pattern is known to be associated with what we call Swiss cheese. We pathologists would like to represent what we see on the slides with food. So sometimes some things have been described with certain food types. So this is what we call Swiss cheese. We call the cribriform pattern. And this is a tubular pattern. You know, the tumor islands are kind of like in tubules. So this is a tubular pattern of adenocystic carcinoma. P63 also would highlight the myoepithelial cells, as you can see here, and the remainder of the ductal cells are negative. So again, this tends to show you that this is definitely a biphasic tumor. The next category after our Dr. Esina biphasic, our next category in which you can classify salivary gland tumors are those tumors that are purely myoepithelial. So the benign version of that is the myoepithelioma. The malignant version is the myoepithelial carcinoma. The next category of our classification system for salivary gland tumors is the excretory duct phenotype. Common examples such as nephroepithelial cyst is under the category of the excretory duct phenotype. The keratocytoma, the mucoepidermoid carcinoma, lymphadenopathy, and squamous cell carcinomas are all under this category of your salivary gland tumors. We'll talk about mucoepidermoid carcinoma very briefly also, because this is the most common malignant salivary gland tumor. Mucoepidermoid carcinoma is the most common malignant salivary gland tumor, usually located in the parotid gland. In the oral cavity, we also see it in different sites. It could occur in the palate, it could occur on the retromolar pad, the buccal mucosa, and on the lip. And interestingly, when it occurs on the lip or on the retromolar pad, it could actually appear like a mucocele. For those of us who are dentists, I'm sure you understand what we mean by a mucocele. So sometimes it could be a diagnostic challenge clinically, because what you're seeing clinically, it looks like a mucocele, which is very benign. It's a reactive process, probably due to trauma. And after doing your biopsy, the pathologist tells you this is malignant. I'm sure you'll be surprised. How can something that looks like a mucocele be malignant? So this is a very good example of such, whereby when it occurs on the lip and retromolar pad, it could present as a mucocele. Histologically, there are 3 cell components in mucoepidermoid carcinoma. There are mucous cells, epidermoid cells, and intermediate cells. We also tend to grade mucoepidermoid carcinoma into 3 different grading systems, such as the low, intermediate, or high grade. The known genetic alteration in mucoepidermoid carcinoma is also known to be citric MAL2 or citric 3 MAL2. And here's an example of a low-grade mucoepidermoid carcinoma. And as you can see the cystic areas in this tumor, and you can see some of the mucous cells in this tumor. This same tumor was stained with P63 to highlight the epidermoid cells, and you can see the nice epidermoid cells, all positive. This is a nice nuclear stain that kind of shows you where the epidermoid cells are located in this tumor, in a low-grade mucoepidermoid carcinoma. Then the last category of our division of salivary gland tumors will be your miscellaneous or indeterminate category. And an example of that is your signet ring cell mucin producing adenocarcinoma. So just to show you some clinical images of salivary gland tumors, this is an clinical image courtesy of Dr. Boku, and you can see the salivary gland tumor involving the parotid gland. Here's another salivary gland tumor. Specifically, this was a pleomorphic adenoma involving the palate, as you can see here. And here's another salivary gland tumor. In this situation, this is involving the labia mucosa, the lip, the upper lip. And this is the tumor when it was surgically removed and sent to us for processing in the pathology lab. This was what it looked like. when it was bisected. And this tumor eventually was diagnosed as a polymorphous adenocarcinoma. So going into our major study today, would be talking about secretory carcinoma, which is our focus in our study. Secretory carcinoma is a recently described malignant neoplasm, and this was identified by Salivary et al. In 2010. Obviously, 2010 is no longer recent, but it's one of the most recent salivary gland tumors that we've identified in the field of oral pathology. Secretory carcinoma was originally named mammillary analog secretory carcinoma, and it was called mammillary analog because of its striking morphologic, immunophenotypic, and molecular similarities to the secretory carcinoma of the breast. So this was said to be okay. It looks just like the secretory carcinoma we see in the breast, and as such, we will call it mammillary analog. However, it's been found that it's actually been noticed and found out that secretory carcinoma tends to occur more in the salivary gland than it occurs in the breast. And because of that, recently, the name mammillary analog was removed, and now we just call it secretory carcinoma of salivary gland. Secretory carcinoma morphologically presents with microcystic, papillary, solid, and tubular growth patterns with variable amounts of intraluminal eosinophilic. Eosinophilic simply means something pinkish. Eosinophilic, homogeneous, or bubbly secretory material. And these tumors have been found to be consistently positive for S100, which is a form of immunohistochemical stain, and also mammaglobin, which is also a form of immunohistochemistry stain. So this secretory carcinoma has been well published that they are always found to be consistently positive for these two immunohistochemical stains. However, recent gene fusions has been identified to further characterize secretory carcinomas. And in the literature, what I found out was that ETB6 is a common gene partner when it comes to the gene fusion or chromosomal translocation, tends to occur between ETB6 and NTRK3, or ETB6 and RET gene, or ETB6 and MET. So the most common of these three actually is the ETB6 and NTRK3. That's the most common chromosomal translocation that tends to occur in this tumor. So the aim of our study was to identify the clinicopathologic features of secretory carcinomas of the minor salivary glands. Now, to point out here, secretory carcinoma typically has been well reported in the parotid gland, in the major salivary gland, specifically the parotid gland. However, there have been very little study looking at the clinical pathologic features of this tumor in minor salivary glands. And as such, we then thought it would be a good idea to study this and also review the current literature and to be able to find out some specific findings associated with this particular tumor in the minor salivary glands. And one of that major thing we also did was to look at possible diagnostic considerations, which we will talk about. So what we did in our matrixal method was that the pathology files from the University of Pittsburgh Dental School and the Joint Base in San Antonio Air Force Base, we went through their records to identify cases that were previously diagnosed as secretory carcinoma or mammillary analog secretory carcinoma between the years of 2013 to 2018. Once we identified such cases, we then went ahead to molecularly confirm them, and by confirming them molecularly, we had to identify the ETV6 gene rearrangement. We had to identify this translocation by FISH, which is fluorescent in situ hybridization. So once we did that, we identified that all the cases that we identified were known to be positive for ETV6 gene rearrangement. If ETV6 was negative, we didn't consider the diagnosis of secretory carcinoma. The cutoff for our positive ETV6 rearrangement was 30% in 60 cells that were evaluated. So we needed at least 10% of the cells to be translocated for us to be able to say, yes, this is positive for ETV6 rearrangement. Cases that were negative were excluded. The clinical information of these cases were retrieved, the age at diagnosis, the gender, the anatomic site, and the size of the tumor. In addition, we also did look at the stains that were performed on this tumor. like the S100 mammaglobulin and P63 stains were retrieved and then recorded. These were our results put in a tabular form for you guys. So we're able to identify 4 cases, even though that might definitely look like a small number, but I told you earlier on, these are very rare tumors located in minor salivary glands. From the literature review that we did, we were only able to identify 63 cases published in the literature worldwide. Only 63 cases were published in the English literature worldwide. So we're able to identify 4 cases. And as you can see, the gender distribution appears to be equal in our series, though very small series. The age range from 23 to 64. The common site is the lip. We have 2 cases on the lip. All our tumor sizes were less than 1 cm. With the stain of S100, they were all positive for S100. Everything was diffusive positive with the exception of 1 case, which was focal. And I will show you that. I will show you a photomicrograph of that very soon. All cases were diffusively positive for mammaglobulin. They were all negative for P63. They were all negative for P63, and this is consistent with other findings in the literature. Then we did carry out our translocation, our FISH, to identify the ETV translocation. And in our study, as in the 4 cases we present today, they all had positive translocation. And you can see here the percentage of translocation. Even though the cutoff was 10%, we were able to identify for example, case 2, all the cells were translocated 100%. Case 1, 96.7% of the cells were translocated. So all these cases definitely went beyond and above the cutoff mark for positive translocation in these tumors. Here's a case 1, the very first case, and this is an H&E, and this is on low magnification. You can see the way the tumor looks like. You can see it has a lobular pattern in there. You can see it all around has a lobular pattern. And if you tend to look in the tumor islands, you kind of see some areas of kind of looks like secretion, areas that kind of have some vacuoles with some secretions in there, like in this area. You can see this pinkish secretion. Also here, you can see this pinkish eosinophilic secretion. It's pinkish secretion. You can see the same thing here also. Let's go to higher power. And higher magnification, you can actually appreciate the vacuoles, the microsecretions everywhere in this tumor island, again with the similar pinkish homogeneous eosinophilic secretions there on high power. This is a stain for mammaglobulin, and you can see mammaglobulin here was positive. This stain is the cytoplasmic stain, so you don't expect the nucleus to be positive. What you expect is the periphery of the nucleus to be positive, and the secretions also are also known to will also be positive for mammaglobulin, as you can see right here. The secretions are positive, and the cytoplasm around the cells will be positive for mammaglobulin. S100 is known to be a nuclei and cytoplasmic stain, and in this particular tumor, we expect the cells to be positive. As you can see here, the nucleus, the Dreos nucleus here, you can see they are all positive. You have this positive nucleus, everything showing red staining. So this is S100 is positive in this case. And this is the FISH, the fluorescent in situ hybridization. And you can see the breaker part, or the breaker part of the probes, kind of showing you that there is positive translocation and rearrangement of ETV6 in this particular case. Here's another case again, case 3, just to show you another example of how this tumor could present. This particular tumor was seen to be, was kind of like in the middle of the duct, in the middle of a cystic duct. As you can see, you can see this lining right there, kind of representing a duct-like area, and you have this tumor island kind of floating in this duct-like area. High power magnification of this tumor. You can still appreciate the vacuoles within this tumor, and within this vacuoles, you can also appreciate the pinkish. You can see the pinkish material which is being secreted by these tumors also. Mammaglobulin in this particular case was obviously positive. Again, you can see that the cytoplasm was positive while the nucleus is negative. The nucleus is negative, but the cytoplasm around the tumor cells, around the nuclei, it's definitely positive for mammaglobulin. S100, in this is the case whereby the S100 staining was focal. It wasn't diffuse like in the very first case. So we tend to report this as being focal because you can see it's just kind of a little part of the cells that tend to be positive for S100. So we tend to report this as being focal, but we still consider it to be positive. And the FISH was also done for all cases, and you can see the FISH for case 3 here. The same explanation as the previous one, and the ETV6 rearrangement is positive in this particular case also. At this point, I'll briefly want to share with you the various genetic alterations that have been reported in different salivary gland tumors. And our study for today is focused on secretory carcinoma, and you can see all the translocations that are positive in these tumors. And as I said earlier on, ETV6 and TRAC3 is the most common or most prevalent translocation to be seen in secretory carcinoma. However, there's a list of other salivary gland tumors, as you can see here, that are also known to have genetic alterations, either translocations or mutations or amplifications. So all this could happen, thereby being the major genetic alteration that results into carcinogenesis or tumorigenesis in these various salivary gland tumors. In our discussion and our review of literature, we were able to identify that the lip was the most common location for secretory carcinomas in minor salivary glands, and not just the lips, but specifically the upper lip. So out of the total number of cases reported in the English literature worldwide, 23 of them, about a third of those cases, tend to occur in the lips. The ratio of the upper to lower lip was 2.4 to 1. So more cases tend to occur in the upper lip than in the lower lip. This is true for other kinds of salivary gland tumors. Other kinds of salivary gland tumors also, it's been well reported that the upper lip is the most common location compared to the lower lip. So this also holds true for other salivary gland tumor. Majority of secretory carcinomas that tend to occur in minor salivary glands are in the same, from the literature review, are shown to have a size less than 1 cm. And this was consistent with our cases also. All our cases were less than 1 cm. Most of the clinical presentation of secretory carcinomas in minor salivary glands, they tend to present as painless raised nodule or mass. So they are usually, you see a nodule, it's painless in most situations. And in most cases also, there were no regional lymph node involvement. In all our 4 cases, there was no lymph node involvement. And the same in the literature review we also did. Treatment for most cases of secretory carcinoma or minor salivary gland is just by surgical excision. And once the surgical excision is done, recurrence, local recurrence or distant recurrence is rare. And the prognosis is very good for these tumors in the minor salivary glands. Now, secretory carcinomas, as I said earlier, one of the things we also did focus on was to look at the diagnostic consideration. As a pathologist, once I get a slide and I look at a slide, one of the things I want to consider is what else could this be? What else could this look like? So that was what we also that's what we also did in the study, to look at other tumors of salivary gland origin that could also present like secretory carcinoma histologically or morphologically. So tumors that shares morphological or immunophenotypic similarities to other salivary gland tumors were evaluated, such as arsenic cell carcinoma, specifically the zymogen poor, low-grade mucoepidermal carcinoma. I showed you a picture, a photomicrograph of this. Intraductal carcinoma of salivary gland. This is actually a very recent entity that was just described last year, microsecretory adenocarcinoma. It's a very, very good mimicker of secretory carcinoma. And lastly, adenocarcinoma NOS, or cyst adenocarcinoma NOS. So I will show you a couple of images for you to compare yourself. So this is a typical arsenic cell carcinoma, and this is a typical secretory carcinoma. Now, The cases of arsenic cell carcinoma that could look like secretory carcinomas are those that are considered to be zymogen poor. So imagine if all this bluish area, if they were absent from this tumor, and you can see these vacuoles, definitely, this would definitely look like a secretory carcinoma. Also, low-grade mucoepidermal carcinoma. You can see the low-grade mucoepidermal carcinoma with the cystic areas, and this is another example of a secretory carcinoma. I'm sure to the unlearned eyes, if you look at this, you will probably say, where's the difference? There's really no difference. So that is why this is considered to be, a morphologic similarity or a morphologic mimicker of a secretory carcinoma. So you can see the low-grade mucoepidermal carcinoma just looks like a secretory carcinoma. Another example again here is looking at intraductal carcinoma. You can see the way an intraductal carcinoma looks like compared to a secretory carcinoma. They look alike, and they could easily be confused with each other. To make matters more interesting, for these two tumors, they also share the same staining pattern, which I would report to you soon. They stain similarly. They are both positive for S100. They are both positive for mammaglobulin. So that could be very confusing for these two tumors. This is the most recent entity that was just described last year, 2019, by Bishop et al. And this is a microsecretory carcinoma. You can see the way it looks like. You can see the secretory areas, and looks very much like this tumor, which is one of our cases here, secretory carcinoma. So again, this could be very confusing to the pathologist who is not familiar with this kind of cases. So you can see both examples here. Now, morphologically, I hope I've been able to prove to you that those tumors could look alike morphologically, histologically. Now let's go to the staining pattern. For your secretory carcinoma, which is what our study is talking about today, we know that secretory carcinoma is positive for S100, it's positive for mammaglobulin, we know it's negative for PCC3, and we know if we do the molecular analysis, we're going to identify an ETB6 fusion. Arsenic cell carcinoma, the zymogen granule poor type, the good thing for us is that it's, based on the immunophenotypic expression, it's negative. So it's negative for S100, negative for mammaglobulin. Also, very good is that new molecular analysis for arsenic cell carcinoma have also revealed that they have a completely different genetic translocation, which is very good. Low-grade mucoepidermal carcinoma, immunophenotypically, is also very different. And also, the molecular analysis, the gene translocation is different from secretory carcinoma. So morphologically, histologically, on your H&E slide, they might look alike. But if a young pathologist is confused and is not able to make that distinction based on just looking at the slides, then the pathologist can decide to order some stains. And by ordering these stains, you'll be able to distinguish this from secretory carcinoma. Now, this is the more interesting ones. The intraductal carcinoma. The intraductal carcinoma, actually, this is not a new tumor, but it's a new terminology. Intraductal carcinomas, these were previously called the low-grade cystadenocarcinomas and the low-grade salivary duct carcinomas. Both of them are now called intraductal carcinomas. If you look at the staining pattern for your intraductal carcinoma, as you can see, it's positive for S100, it's positive for mammaglobulin. So this could be very confusing to a young pathologist who is not in a big institution where molecular analysis can be performed. If you're confused and you're thinking, oh, is this intraductal carcinoma? Is this secretory carcinoma? Okay, let me stain for S100 and mammaglobulin. After all, it's been reported in the literature that these two stains are known to be positive in secretory carcinomas. And you go ahead and you stain for these two stains and they come back positive. And you say, okay, I'm going to sign this out as secretory carcinoma. But you could be wrong because this is not secretory carcinoma, this is intraductal carcinoma. In bigger institutions, they might go ahead and they could perform FISH. If they go ahead and do their FISH, the fluorescence in situ hybridization, they will be able to identify that the translocation in this particular tumor is different from the classic ATV6 translocation. And that might just be the saving grace in the ability to be able to make an accurate diagnosis. And the new category of tumor here, which is the microcystic, one, as you can see here, also could be positive for S100. So that could be a little bit confusing, but we know that the translocation is very different from your classic ETV6. So why are we talking about this translocation, genetic analysis, molecular analysis? Now, none of the morphological immunophenotypic mimickers are positive for ETV6 rearrangement. Thank God. So with that, we can be able to make that diagnosis accurately if we then go ahead to do the ETV6 rearrangement. But again, the truth is that this is not readily available in all institutions. Most places don't do FISH. Probably the big institutions, the tertiary institutions, are the ones that have the ability to be able to perform the molecular analysis. So with the advent of molecular analysis as a routine investigative and ancillary tool in clinical histopathologic diagnosis, FISH for ETV rearrangement is suggested to be performed before rendering a diagnosis of secretory carcinoma of salivary gland. Now, beyond just for diagnosis reason, the application of molecular diagnosis is critical, not only for making a diagnosis, but also providing information on prognosis, and also it helps to guide therapy. And I would like to conclude here, because I think this is the hallmark of everything I'm presenting today, is that with the advent of molecular diagnosis identifying such translocations, we can now go ahead and provide precision medicine. We can then go ahead and provide targeted therapy for our patients. And this is a very good example for this particular tumor. Patients that are diagnosed with ETV6 and TRAC3 fusion, secretory carcinoma, they've been shown to respond well to tyrosine kinase inhibitors, because this NTRAC3 is a tyrosine kinase, gene, and as such, tyrosine kinase inhibitors such as entrectinib have been shown to be very effective in these tumors in patients that have secretory carcinomas. So if you treat them with this form of targeted therapy, the tumor will literally shrink, and that could then help reduce the morbidity associated with the surgery. Now, I know this might not play a big role in minor salivary glands, because earlier on I said the size of the tumors are usually less than 1 cm. But again, this particular secretory carcinoma, as I said earlier on, it's more common in the major glands, in the parotid glands. And when they are in the parotid glands, they could have a very big size. They could be as big as 10 cm. So the ability to be able to use such targeted therapy would then be very important, because that could help reduce the size of the tumor, and that will reduce the morbidity associated with such surgery. Another example, again, with this same tumor, is that tumors that are known to be positive for ETV6 RET fusion, secretory carcinomas, could also benefit from the RET targeted therapy, such as the Bloom 667 and the LOX-O 292. These are still under clinical trials, and there have been very, very good responses with this form of therapy in treating patients who have RET fusion positive tumors. And another last example here is the use of ETV6 MET fusion. These patients can also benefit from the c-MET inhibitors also, like the carbazolmetibine has also been shown to be effective in treating patients that have a MET fusion. And that will be the end of my conclusion of my presentation. These are my references. I'd like to thank everyone for being here to listen to this presentation. And I would like to acknowledge the AT Steel Research Institute for funding this work, and my collaborators at the University of Pittsburgh Dental School, Dr. Somersgill, and also at the Lackland Air Force Base, Dr. Tyler, for providing this tumor cases, and we were then able to carry out this molecular analysis on these tumors. At this point, I would like to take any questions from the audience. Thank you. if you have any questions, please, I would love to take your questions now. Any question?
Dr. Owosho?
Yes.
This is Dr. Bradbury here. You mentioned early on that you were going to exclude tumors with lower than 10% translocation. Did you find any such? Or were these only four that you actually found in the bank?
Yeah, very good question, sir, Dr. Bradbury. Yes, we were able to identify 2 cases that were suggestive of that diagnosis. But when we did the translocation, they did not meet the threshold. So we had to exclude 2 cases from the total 6 that we identified.
So what were those likely? Do you have some idea of what those really were?
Then, like I said, we talked about the possible differentials, the possible diagnostic considerations. The truth is that those could probably, most likely, they probably would have been intraductal carcinomas, because the intraductal carcinomas, they look alike, they also stain alike. So probably those cases were intraductal. We did not investigate further because our focus of our study was kind of limited to this secretory carcinomas. But yeah, but I believe that would be something that we could look into to then know what that diagnosis was precisely.
Hi, Dr. Owosho.
Yes, sir.
How are you?
Fine, sir.
Yeah, just my question. As you mentioned, this kind of carcinoma is discovered in 2010. So
Yes.
before that time, what's the provisional diagnosis for this kind? I'm sure there are some patients, they were complaining of this kind of cancer.
Yes.
So do you have any idea what's the provisional diagnosis for those patients?
Yes, sir. That's a very good question, Dr. Shitty. So the truth is that when the community, the oral pathology, head and neck pathology community identified this particular new tumor, most people then went back to their archives, what they had previously diagnosed as a different entity, and what they had to do was that they had to revise their diagnosis. They had to go back there, look into their archives, and they had to do some research work to basically to re-diagnose those cases. So in the past, they were initially called actinic cell carcinomas, the zymogen poor type. That was the most common diagnosis that was rendered for this before the diagnosis was made. But now that we know that it's a completely different entity based on the histologic, morphologic, phenotypic, and molecular, that it's completely different from actinic cell. Now, going forward, this is now what we call them anytime we see this. And most of the time, we would perform stains. And even after the stains, if we're not convinced, we will then go ahead and do the molecular analysis to be able to arrive at this diagnosis.
Oh, okay.
Okay. I had a question about these mucoceles that look identical to a mucocele. Clinically, how are we to know who to send for a study?
That's a good question, Dr. Cohen. Yes, so especially with low-grade mucoepidermal carcinomas, they could, especially when they occur in the lip or retromolar pad, they could just present like a mucocele. And I know most dentists, once they convince it's a mucocele, they probably won't send it into the pathologist. But it's good judgment to always send anything you remove. If you're biopsying anything, it could even be an excisional biopsy, to always send it, even though you're 99% sure it's a mucocele. It would be nice to send those in to the pathologist just to make sure that yes, it's 100% mucocele. There have been a couple of examples that they turn out to be something else entirely. So sending it to a pathologist nearby, especially an oral pathologist, because the truth is that most general medical pathologists, they're not familiar with the oral pathologist. So sending to an oral pathologist would be a very, very good advice.
Thank you.
Yes, sir.
Okay, Dr. Owosho, I'm sorry, another question.
No, that's fine, sir.
Okay, so based on the ETV6, this is main feature only for this type of cancer, or could be seen also in other kinds of carcinoma? So
That's a good question. ETV6 translocation is not specific to secretory carcinoma. Like I said earlier on, we could see secretory carcinoma itself in the breast, and there are other tumor types that also have the same this translocation ETV6. Even though it might be ETV6 translocation, the gene partner might not be NTRK3. The gene partner might be a different gene partner, but if it's just the ETV6 alone, there are other tumors that have been described to also have this ETV6 translocation. So it's very possible there are other tumors out there, but the good thing is that when we talk about salivary gland tumors, salivary gland tumor as a whole, even though we have about 30 examples of them, this is the only tumor that has ETV6 when it comes to salivary gland tumors. And that is why when you send a specimen to a pathologist, the location, the site of that tumor is very important. So yes, so salivary gland tumor, this is the only tumor that has ETV6 in all salivary gland tumors. And even in the oral cavity, if you think of something from the oral cavity, other tumors of the jaw in the oral cavity, this is the only tumor that has ETV6 translocation.
Oh. Yeah, it's exciting. Congratulations. It's good. It has a good study.
Thank you, sir. So if in the absence of any other question, I would like to share with you our ongoing research work, which I'm currently doing, and I'm fortunate to have funding from the AT Steel Research Institute. Currently, we're looking at the molecular characterization of pleomorphic adenomas and adenocystic carcinomas in Nigerian patients. In that part of the world, no form of molecular characterization has been done for these categories of tumors. So it's of interest to me. Some of you might know I'm originally from Nigeria, so I would like to see what's the prevalence, and do we actually have the same molecular characterization for these tumors in those patients. So that's something we're currently working on. And that new study we're also working on is we're evaluating the use of evaluating BRAF mutation in ameloblastomas. Ameloblastoma, it's a benign odontogenic tumor of the jaw. And as a dentist, this is something I'm sure we know of, and we were probably taught multiple times in dental school, ameloblastomas. Recently, the mutation of BRAF was identified in ameloblastomas. I was fortunate enough to get 59 cases, distinct patients with ameloblastomas, and I would like to check for this BRAF mutation in these cases and hopefully publish the findings of what we identify. Now, what's the usefulness of this? Is it just for molecular reasons? No. Actually, there's a therapeutic benefit to this. As some of you are familiar that BRAF, the name BRAF is, when you think of BRAF, you think of melanoma and other tumors. And there are known anti-BRAF medication, BRAF inhibitors, that if we're able to identify the same mutation in these tumors, I am thinking in the future of a possible clinical trial whereby these patients could be provided with a BRAF inhibitor and see if it will help these patients in providing therapy for these tumors. Some of you are familiar with ameloblastomas. When they grow, they could be very massive. They could be very massive, very destructive to the jaw. And as such, just doing surgery alone would leave a very big morbidity to these patients, because in most cases, you would have to remove the entire jaw to treat these patients. So if you can find an alternative form of therapy with the use of the BRAF inhibitor, that would help reduce the morbidity associated with such surgical treatments. So these are ongoing works that we're looking at, and hopefully I'll be happy to come back to share these presentations with you once they are accepted in peer-reviewed journals. Okay, In the absence of non-linear question, thank you very much for attending this presentation. Thank you for your wonderful, informative, interesting questions. Thank you very much, everyone.
Thank you, Doctor.